Okay. So yeah, my name is Aabid Sharif. I grew up in Chennai in Anna Nagar, and the St. Peter's College is right there in Chennai. I miss many things in Chennai, but I was there a few years ago. I'll be back there soon. So we'll dive right into it. A lot of my career has been in moving from uh, um, biological engineering to moving into leveraging AI to solving biological problems. Back in the day, we used to call it computational biology. Computational biology has always leveraged machine learning for solving many problems. Many of you are already aware about that, but AI in itself is unique, and I'll talk a little bit about that in this talk. So there's lots of parts in here that I can share will be more like a learning opportunity. But many things are also focused on the research work that I've done, but will not cover some of the recent work that I've been doing at Citrus or at Opal Therapeutics. I've kept that for later. But for now, we'll just talk on some of my past work that I've done at Grassroots, at Monsanto, Bayer. So you'll see some work in crop science as well. But it really ties into the field of biotechnology. Okay, so diving in. Yeah, I did my little background about myself. I did my BTech at AC Tech at Anna University. I then we did lots of internship at NCBS in the space of neuroscience, in genetics. Did my PhD at Carnegie Mellon in Pittsburgh. Worked at a spin-off out of Duke University called Grassroots that focused on gene expression. I'll talk more about that. Then worked for we got acquired by Monsanto, now Bayer, and from there went on to work for a CRISPR company, a Broad Institute founded CRISPR company called Pairwise, where I was head of data there for about 5 years, and then decided to start my own company. We'll talk more about that. So the talk outline is pretty straightforward. I'll talk about AI, ML, the phenomics work that I've done primarily in high throughput phenotyping, the work in genomics, and how generative AI could really lead to lots of design opportunities, and finally how we apply that to gene editing, which is making small changes to the genome to have significantly large changes that would benefit in the healthcare space as well as in the agriculture space. I'll give an example in agriculture. Okay, we've heard this word digital transformation a lot. Really, it started with software. Software started to change the world significantly, make things more efficient, faster, and improve the quality of how we did work. That really happened also in the biological sciences space. It just started with simple software writing, but then real true biology started happening where you could do things like blast sequencing. You could use apply blast. You could apply the technologies for sequencing, like what Illumina does, has a lot of underlying machine learning algorithms underneath it. And with the advent of now AI, that is really the excitement around is really around generative models, what we call generative AI. Many of you are probably already familiar. Many of you are using these tools already. But yeah, I'll talk a little bit about the nuances around this, about what exactly is generative models and how that would apply to life sciences. Okay, so I'll start a little bit about machine learning. The reason why I have this slide in here is because it's still not very obvious to many people what exactly machine learning is. So, but I'll share it. I'm sure many of you know, but for the sake of bringing everyone to the same table, I'll just share it. For traditional programming, you talk about a data input, right? You say, I'm going to provide some data. You have a program that you provided, and then the computer will spit out an output. But in this context of machine learning, the paradigm shifts a little bit. You provide the data, you provide the output as well, and then you ask the program to figure out a program, a model that can actually create a mapping between the input and the output. So every time you have new inputs, it can already create the outputs. So it becomes like this, but then the way you design it is you don't design the program gets designed by the computer. And so I think that in itself created this paradigm shift creates a change in which how people think about things. um, in the standard ML literature, we call this training data. Okay, so now data science in the AI world is really you go from data is now called training data, and the problem that which is the tool to solve the problem is really the model that we are building. And this is what a lot of AI data scientists spend trying to build a model that can solve a problem, which is trying to take an input and then give you the output that you desired. It could be designing new drugs, it could be designing new sequences, it could be as simple as improving the quality of sequencing, or doing predicting if a machine is going to fail. So it could be many, many different kinds of problems. And we've seen most recently in language generation and natural language processing with ChatGPT. Okay, my computer just crashed. Give me 1 second. Let me open up.
So can I share these slides, Abid? Is it okay?
Uh, it should be. I just got it fixed now. It should be
Yeah,
working
yeah.
now. Is it working?
Okay, okay, okay.
Let me continue here. Okay, so the general approach is trying to create, and this is if anyone's trying to get into the building ML programs within your labs or within your groups, the most important thing is to set this kind of cycle up, creating learn, design, test cycles. So you have a problem that you're trying to solve. You want to learn from some data sets that you create, what works, what does not work, and you'll see that what does not work is as important as the word data that actually works. It could be again drugs or sequences or whatever it is. And then you have your program that will that will learn based on the data. This is the model basically, and it spits out new sequences that you can then test out in the lab or in the or in silico, that then you can keep doing this chain of learn, design, test to improve. And this is really the standard now in all of pharmaceutical industry and biotech industry. Everywhere you see these kind of learn, design, test cycles are being set up. I mean, I would argue that this kind of format is true anywhere else as well, outside of any industry. Even marketing has now adopted this sort of approach to improve constantly. How do you improve better approaches to marketing? So you the general format is you start to ask what exactly is the question you're after. Like for example, what DNA sequence do I design? Then you build your model. The old school models are more mechanistic models, and the new modern ones are more statistical, which are based on machine learning. The very often in life sciences we require high quality data sets, but we don't necessarily have to build expensive data sets. You can take advantage of lots of public genomic data or public data sets that you can use to train your model. And I think great ideas just start from there. It's really about figuring out what is the question that you're after, what is your niche research area that you care about, and then building this sort of structure this way. And then finally, validate the model by doing testing. And this becomes your very interesting data that can show you that your approach works or does not work. And the moment you see that there's not much improvement or you don't see anything working, you can actually kill the project and then work on a different problem. So that's you want to fail fast so you can see what actually appears to work and what does not work. Or you might say this is an important problem enough that I really want to dedicate my life towards, and then you work on building a model that you say, hey, I'm really going to try and understand the biology. I'm going to try and build the model in a way, an AI model in a way that actually understands the problem and can address the problem and really improve the state of the art. Um, so I've used the word mechanistic and I've used the word generative. So I'll explain a little bit to know what I mean by that. So mechanistic is basically we've been studying lots of gene targets. We've been studying, we've understood how proteins work. We spend a lot of this time understanding, but we take that human knowledge of how we understand various things which we call reductionist studies, or it could be at some level systems biology studies as well. We understand what gene targets are interesting, what are useful, what are part of pathways. You understand how the chemistry works. You understand how the physics works. You put all of that together into a nice model that says this is how I think the human biology works. This is how an organ works. This is how this drug is supposed to address this disease state. And you build a model and you use that model. The model will have some sort of accuracy around it. It can address some cases. It'll help you design a few set of compounds that can be useful for your research program. But in the generative case, which is what you're seeing with tools like ChatGPT and others, is where you provide the entire different sets of data sets already. So let's say the example, in a simple example, is you're trying to figure out the difference between oranges, grapefruit, apples. And you provide lots of different images of grapefruit, apples, and oranges. And you tell the computer to figure out what is interesting about an apple, an orange, and a grapefruit. And once it learns what is interesting about it, you ask it to then generate its own, you generate your own apple, you generate your own orange, your own grapefruit. And in the case of biology, this could be an example of certain class of proteins, right? It could be kinases or it could be something else. So what you're trying to say is now that I've given you, fed you a huge amount of data around these kind of proteins, generate new proteins for me. So you can start to see how this sort of approach would lead to design of new molecules. Um, I actually didn't start my career in once I finished my PhD work, and I did that in image generation, generative AI around images. Computer vision is where actually generative AI started. Uh, um, uh, that's where I think the algorithms were designed. But then once I finished my PhD, I went to work for a startup called Grassroots, where we were trying to design novel ways in which the roots of plants could absorb the nutrition better from the soil and then increase yield. So, or increase any other different interesting traits that are relevant to many things that we're trying to work on. So, I started with having high quality data sets for how when you test a particular promoter or test a specific gene, what would happen to the roots? Do the roots change in length? Did it improve its properties? So I started my career actually in industry working on doing research and building this high throughput, high quality data sets. And this by far, I think, is the most important gap right now. Is you see in the recent years, there's lots of talk around AI algorithms in life sciences, but building high quality data sets, I think is truly going to move the needle. The lab work that we do in building consistent quality data is going to be very useful and will be the paradigm shift in my opinion. So I started working on building these data sets, but then I had no experience, as much experience in genomics. Then I started trying to dive into it a little bit more. What is the how what are the different parts of a gene? There's a promoter, there's actually protein sequence, but there's also the other parts around it, the 3' UTRs, the 5' UTRs, the introns, what we largely call junk DNA. I really wanted to understand how those junk DNA can contribute to changing expression levels so we can get to a state of design. How do I increase this gene? How do I decrease it to a certain level? How do I say that this gene should be expressed in only certain tissues, not all tissues? I want it to be expressed at this time point and not at that time point. So can you come up with that sort of design of DNA sequences that will lead to what we call designer biology, right? And that will have an impact in healthcare and life sciences, in broadly many areas, including industrial biotechnology. So what was interesting about gene regulation is that there are many different types of data sets that you can acquire. And many of you probably work on many of these areas already, but this whole thing gives you an idea of how a system works and how things are regulated. It's clearly a systems biology problem. You got to really look at it as a system. And what better to understand a system than leveraging AI tools? So I showed you earlier about this is a quick cartoon that I built around what a gene looks like and the various areas around the coding region that actually control expression. Um, and so one of the things that I did is to try and figure out how do I create a mapping between sequence and expression levels. So, and I'll share a little bit more about why that is useful. So before you do that, you got to try to acquire high quality data sets, high quality RNA-seq data sets that are tissue specific, right? You want to be able to say, in this case, it's plants, but if you're doing since this is a personalized medicine conference, you can imagine acquiring high quality organ specific tissues or getting single cell data around very clearly understanding epithelial cell layers or exactly the different types of cell types and creating high resolution RNA-seq data sets. Back then, we didn't have a single cell RNA-seq. We had to leverage on tools like laser capture microdissection that can extract out tissue layers, and then you can build your high resolution data sets. Again, we acquired lots of different data sets across many different tissues, in this case, corn. And I won't go into much detail, but we were able to clearly say, create, you need to cluster all the data. You can clearly see the, the differences in expression across the different genes in the various tissues. So one of the things that we were able to do is to say, okay, this is one specific organ, this is another specific organ, and this is another specific organ. So when you start to do clustering methods of this sort, so now we have, if we look at those genes and extract out those junk DNA around those genes, you're able to then say, now can I use AI to start learning about what is interesting about those sequences? So one of the things that we did was to take those training sequences. Let's say this is leaf data, this is root data, and you can apply the same concept to other areas as well, in the human space as well. You can use mouse models or any other data sets that you can use to say this is specifically eye tissue, or this is specifically heart tissue, or whatever that is, and then take those sequences and then build our generative models that could learn from those sequences. What exactly they're learning is highly statistical, right? They're trying to figure out interesting correlations that we don't really understand. But then what was interesting is that it started to pick out interesting motifs, which we now know are those motifs are really transcription factor binding sites in these sequences. So we were able to design de novo. De novo means you take these sequences and you blast it to, you do look a similarity check to anything in nature, you find zero similarity, but they work exactly as intended. If you want an enhancer that will increase expression by 50%, you can exactly design that sequence, all designed by the computer. So, the several patents were awarded for this work. I have at least about 10 patents around this space. The recent one that was awarded to me about a few months ago, that for an AI that I built about 10 years ago. So the AI is still generating novel sequences that is being used in many different products in the space of life sciences, space of pharmaceuticals, just to in the space of gene expression. Again, I won't talk about, I mean, I can't really show the data. A lot of the data is proprietary. We're waiting for some of the publications to come out. But I think this was really around generative AI for modulating gene expression. I further asked the question, can we apply generative AI to another popular problem called codon optimization? Many of you have probably heard about this. When you design a sequence, or when you're working with proteins, let's say you're working with a CRISPR protein, or you're working with you take a sequence from bacteria, you study about it, like, or many of the CRISPR molecules is from bacteria. But how do you make that work in humans in a way that it doesn't trigger silencing or trigger rejection? How do you make sure that foreign sequences actually work in the host organism? And the way usually you approach that is codon optimization. And I applied the same approach of using generative models to coding sequence optimization as well. How do you change the codons in a way that a foreign sequence now is understood as a host sequence? And we built one of the industry leading algorithms that the algorithm itself was many of these are actually available with many like these cloning software and cloning tools. But one of the things we built was to make sure that we can, again, say, I want increased expression here. I want to be understood in this organ and not that organ. So that designer type of mindset is what we were able to do with these codon optimization efforts. Um, Now the question is, we have talked so far only about DNA sequences. Can you apply this to amino acid sequences? And we did the same thing for targeting peptide as well. You can design novel targeting peptides that you can say, hey, I wanted to go to not just this specific tissue type, but can you say a protein should go to a specific subcellular organelle? It needs to go to the mitochondria, it needs to go to the nucleus. Can you design the targeting sequences that can go to specific areas? Now, a lot of the work that I've done here, you understand these targeting peptides don't form 3D structures. So these amino acids do not form 3D structures. They're just the targeting sequence that say where it should be localized within cells. But it was really nice to see that the same tools that we applied for DNA sequences to back up, we applied what I did for my PhD in the vision space can be applied to DNA sequences, can also be applied to amino acid sequences. And this was work that we did in 2015. In 2017, Google worked on DeepMind. I was no longer working in the protein space, in the AI space. I moved on to another company. But if you know, the same concept was applied to protein sequences, which most of you know is AlphaFold. It was made by Google DeepMind. I think the difference here in AlphaFold versus what I had done is that I I here they have to consider structure, 3D structure, dynamics, and further additional types of things. Whereas in my case, I didn't have to worry about structure. It's really about trying to figuring out how these amino acids work together to do targeting. It's mostly motif discovery. If you had to give it a name. In 2018, I joined as a head of data and genomics at a company called Pairwise. It's a Broad Institute-based company, but that was trying to make using CRISPR to to make small changes to the genome of plants to make cherries pitless or make some new exciting fruits and vegetables. So we were building a whole portfolio across produce and making changes to plants to bring very exciting things to the market, where it's not about corn, right? It's more about fruits and vegetables that we can eat. You make one base pair change, and you can make a cherry seedless, a pitless. So it doesn't. Kids don't have to choke. You don't have to wait. You can eat the whole fruit and you're happy. And really, you're not making in many of these concepts. You're not really making these seedless. What you're truly making is the seed has an outer shell called endocarp, and you're really making that endocarp softer, so it's more edible. So you give the impression in your mind that it is seedless, but truly you're consuming the seed as well, which is in fact healthy and nutritious. So those are the very interesting concepts that we worked on. We talked about making mango seedless or making the seed a little bit softer than or more edible. Almond is sort of a seed, right, that you can actually eat. So how can you make something an almond inside a fruit that is more edible? So very interesting things we read. But our first product was something called super lettuce. We took mustard greens, is a very popular food in India. We took mustard greens, and if you eat mustard greens as it is without cooking, raw, it's very pungent. It has a raw wasabi-like flavor, taste. but many people don't like it. In Indian cooking, we boil it, very nutritious, and we eat it. But many people want to eat it as salads. And so how do you deal with the pungency? So we made about 18 changes to the genome of mustard greens, and we have to deal with other problems because we have ploidy and all that. But we were able to take that, and we were able to launch this product in within 4 years, start to finish, idea to product in the market, 4 years using CRISPR, using all the advances in AI and genomics. We were able to design the CRISPR molecule. We were able to deliver the CRISPR molecule. We were able to figure out the changes we need to make, all because of leveraging AI technologies to get to where we need to get to. And this work was published uh, uh, um, about a couple of years ago. So with that, I'll just, and I can go on and talk about, lots of details, but, I just wanted to keep it very high level. Really thank my work that I did at Carnegie Mellon and at AC Tech that laid the foundation and broadened the scope, or the biotechnology program that broadened the scope of understanding of life sciences, and the various companies that I worked at. With that, I'll stop and maybe share one key thing that really differentiates in how I think what enabled my career and helped me a lot is really about, is really understanding the domain knowledge, is spending the time to really think about the way, if you can't just apply software or AI to a new tool and say I'm going to win. You have to really dive in, spend a lot of time with experts. You yourself have to become the expert in protein science, gene expression, in phenotyping, in whatever area that you're working on. Neurodegenerative diseases or whatever that is, right? You got to really spend your time understanding that, and then be able to make an impact in that space. With that, I'll stop and I'll take any questions if there are.
Thanks, Abid. Thanks for your beautiful speech. So you were working on neurosciences and you totally shifted your career. Is it right?
Sorry, can you repeat that?
Sorry, like yeah, you started working on neurosciences. Is it right? Like then you totally shifted your career.
Yeah, so I'd say I've broadly been in life sciences. So I went from neuroscience. My PhD work was in cancer biology, and then my work in industry started with plant biology. As you can see here, a lot of my talk was plant work because this is my own research. And then now I work in women's health. Opal Therapeutics is a women's health company that I advise. Built the entire AI program there, and the my own company, Citrus Oncology, is not even in life sciences. It's a healthcare company. So anyway, I didn't want to distract it and talk in all these other areas because I had a lot more content to talk. Maybe next year or the next time you have this event, I'll share more about the work in women's health and healthcare.
Surely. So we'll keep in touch. So the session is open for questions. Is there any questions from the audience? So you can put it in chat. Audience, is there any questions? Yeah, there's a question from Dr. T. Gopalakrishnan. What are the limitation of current generative AI models in replicating biological dynamics, and how can these limitations be addressed to improve the fidelity of simulation?
Yeah, this is a really good question, and it's something that I have struggled with for a very very long time. And just some nuance around this question, like, and please, Dr. Gopal Krishnan, tell me if I'm understanding incorrectly. People when they design, like what AlphaFold stuff is generating static structures. It's not a protein is a moving, living, breathing thing. So how do you how do you really model something that's like really through MD simulations is really is the way you understand. And MD simulations is a mechanistic model. How can AI really try and make a difference in that space? And what is the current state of generative AI models? in those space. So I have been some of my friends have also started companies around this space. Trying to model proteins is a very very hard problem. So I truly think replicating biological dynamics is how we're going to improve the accuracy of these models. Is really how we think that But I would say that we're not there yet. I haven't seen any clear work in this space. So if you look at video generation algorithms where they use things like stable diffusion, I think using that space, that class of algorithms for video generation, which is something more dynamical, and applying that to protein work, I think is really where I think the sweet spot lies. So I think we're going to be there, but we're really we're really going to have to adapt the existing algorithms to meeting protein work. And I got a lot. There's a lot of I don't think it's just software. They also have to be hardware related. So with the new NVIDIA chips, I think we're gonna be able to do some things in that space. But yeah, I hopefully I've shared some insight there. But yeah, it is I think an area that requires significant amount of new research.
So the next question is, what role can generative AI play in predicting toxicological profiles of drug candidates and helping to streamline the drug discovery process?
Yeah, so this problem is actually very near to my heart. It's one of the reasons why we built Citrus Oncology to address from a healthcare from a care delivery side. How can we address side effects of many especially cancer drugs? It's Cancer drugs have significant side effects that are which indeed show up as toxicities. But yeah, I think in the drug discovery space, this is something that there is a standard toxicology databases that people do testing against, but there's not a real understanding around around how this would influence various organs inside the body, or what that would mean. So I truly think that it is a problem that only generative AI can solve, and it's very difficult to just do a scan and match against databases and say, oh, there's no database match for what I've designed, so it should be good. Because when you provide a drug, you're creating a change in the system completely in the human body. And so we really need to build, I think, lots of foundation models of how many things interact together. And foundation model is a different way of really saying generative model, but you consider how things work from a first principles perspective, and you try to understand how. So I truly think generative AI is the only thing that can do a good job at predicting toxic toxicological profiles. Although I will say, since I'm more on the healthcare side and less on the drug discovery side, I've not kept up with literature, but I do think there's lots of things that you can do there in predicting toxicological profiles and being even more accurate.
So the next question is, what role can AI play in identifying the molecular signatures of disease in patients, and how can the generative AI help tailor therapeutic interventions based on these signatures?
Yeah, again, I think if you look at what AI has done in life sciences, especially in the diagnostics space, uh, this is the most natural fit again for AI, because what AI does is really good is at doing is at profiling, right? It's trying to say what is unique about this group of people. Like you see clustering algorithms, like what is exactly unique about this, that, and it's really developing a signature. So if you look at what a GWAS is, when you're trying to do genome-wide association studies, you're trying to find how a disease is connected to certain markers in the genome. Then it's really applying a very rudimentary model. You're really applying a linear model in the back end. So what AI can do is to model nonlinearity. How can a combination of all of these molecular signatures, when combined together, can say, okay, this is a certain disease phenotype of a patient. So I truly think that the best natural fit is is AI in detecting molecular signatures, and this work has there's a tremendous amount of work in this space with many diagnostic companies identifying market discovery. I think is really AI, leveraging AI. And the second part of the question on, can how can generative AI tailor therapeutic interventions based on these signatures? It's this is the all the diagnostic companies. That's their dream, right? How can we use a combination of diagnostics and therapeutics together simultaneously, work hand in hand to really saying when a patient should be applying this therapeutic and when they should not be. So yeah, absolutely. I think this is another really wonderful space for a scientist in the group to working on. But it does. I'm sure there's some literature as well around this space, but I'm not up to date in that area.
The next question is, what ethical concerns arise when using AI to manipulate biological system?
Yeah, I think to me the way I look at AI is less to do with. Um, I mean, yeah, I think there's I think it's hugely overblown. I think the concerns around AI. I mean, if you apply AI to military or in other areas, I understand this real ethical concerns. But with to me, AI is solving three broad areas within life sciences. You are either accelerating discovery, that is, you're making a process much faster, which was usually slower. So it's speed, or the other area what AI is doing is really to improve quality. So what would take if you had to screen 100 drugs, you can now only have to screen 10 drugs because the efficiency of design is much faster. So improves quality. But the last case is improving efficiency. You can do a lot more work in a smaller amount of money pool. So it's like it would help with the resources. So to me, I really think that AI solves these three areas in life sciences. And the ethical concerns around designer AI is really around, should we be doing this in the first place? Should we even designing drugs in the first place? Should we be even applying gene editing for people to be able to design their own babies? So I think that is where that is not an AI problem. I think that is a general problem where people should be asking clear questions: whether should we be doing this in the first place, whether we should be applying AI to manipulate biological systems. I think is yeah, I think is really is I mean we are just learning and making things more efficient.
From Dr. Yashoda, please discuss about AI tools for gene sequencing of a pathogenic organism in agriculture. Can
Yes. Yeah, so again, this is I'll be honest. I've been a little far removed from I got to admit what I don't know. But so some of the work that I've seen, with AI tools in agriculture, people have really spent a lot of time collecting. I think it's less to do with AI and more about collecting vast data sets of what is in our soil, what is really there, why do some places plants grow really well. Is it means not just the altitude, right? It could be, it's not just the rain, it's not just the weather, but the soil itself has certain characteristics. And understanding the microbiome in the soil has been a vast, exciting space. Maybe not as exciting in the human health area, but in agriculture has been a very, you can really show increase in yield when you use a different type of soil type or soil content with microbes. And that really has to do with gene sequencing, of these rapid gene 16S sequencing of microbes that help people understand. So I really can say about, I mean, AI tools specifically around gene sequencing for pathogen, but I can say that this space has a very exciting future. But I will tell one caution though, just around this area from a more a business perspective. there is a theoretical limit of what microbes can do to changing plants or agriculture, to improving yield, or you do at some point you do need to change the plant's DNA itself to take on new characteristics. You can protect it with microbes to some extent, but beyond that, if you want the plant to grow taller or stay resilient to climate change, then you are going to have to change the sequence. So it's going to be a combination of uh, addressing what's in the soil and addressing the plant itself.
Thanks, Avid, for your wonderful